In this video, we're going to talk about DNA replication. We're going to focus on the processivity rate and fidelity of DNA replication. DNA replication is a process of replicating the cell's genome, and this produces two identical DNA helices. Each one has a new strand and an old strand. Some of the main points about DNA replication is that it is semi-conservative. Each strand of DNA acts as a template to synthesize the new complementary strand, and is synthesized in the 5' to 3' direction. DNA polymerase Pol3 is the enzyme used to synthesize the new strand, and it requires a primer. The replosome is the replicated machinery that moves DNA along at the replication fork. It consists of DNA polymerase, the primase, the sliding cam, the single-stranded binding protein, clamp loader, and helicase. The highlighted strand is known as the lagging strand. This strand is synthesized discontinuously in the 5' to 3' direction away from the replication fork. This strand is synthesized in fragments because as the replication fork moves forward, DNA polymerase must dissociate and reattach on the new DNA. The fragments are known as the Okazaki fragments. The highlighted strand is known as the leading strand. The leading strand moves in the 5' to 3' direction towards the replication fork. The leading strand is synthesized continuously because the DNA polymerase is moving in the same direction as the replication fork. DNA replication begins with the enzyme helicase. Helicase moves the replication fork forward by unwinding the DNA, leaving single-stranded DNA regions. These regions are coated by the single-stranded binding proteins, which prevents them from recoiling. Topoisomerase is the enzyme that acts on the duplex in front of the replication fork to prevent supercoiling. Primase makes an RNA primer that is complementary to the template. DNA polymerase, also known as Pol3, extends the primers by adding nucleotides to the 3' end of the existing DNA strand. DNA polymerase 1 removes the RNA primers and replaces them with DNA, and ligase seals the nicks that remain after the RNA primers are removed. Processivity is known as the number of catalytic turnovers per binding event. That is the number of nucleotides incorporated before DNA polymerase dissociates from DNA. DNA polymerase 1 has a processivity number of 10 to 100 nucleotides, while DNA polymerase 3 has a processivity number in the thousands because of the beta sliding cam. The beta sliding cam is a ring-shaped protein that encircles DNA polymerase of the lagging strand and keeps it from falling off when it restarts DNA synthesis at a new Okazaki fragment. Fidelity is the frequency of errors. Think of fidelity in terms of someone being faithful to the spouse. When someone is faithful and doesn't cheat, it means they have high fidelity. When someone is not faithful and cheats, that means they have low fidelity. Back to the case of DNA replication, fidelity is high due to the 3' to 5' exonuclease activity of DNA polymerase 1 and DNA polymerase 3. DNA polymerase 1 has a net error of 1 error per 10 to the 6 to 10 to the 8 nucleotide additions. Pol3 has the same fidelity. Rate is the number of catalytic events completed in a period of time. Now we're going to compare DNA polymerase and RNA polymerase, which is the enzyme used in transcription. Transcription produces a complementary RNA using a DNA template. This is completed by the enzyme RNA polymerase. RNA polymerase is the enzyme that produces the RNA transcript using DNA template strand. Processivity is lower than DNA polymerase because RNA polymerase synthesizes its own primer. RNA polymerase also has a lower fidelity than that of DNA polymerase. RNA polymerase has an error rate of 1 error per 10 to the 4th nucleotide. It is error prone. The rate of RNA polymerase is approximately 50 to 90 nucleotides per second. This is an example of an experiment done to measure the fidelity and processivity of DNA polymerase in yeast. The DNA polymerase has the ability to replicate past a dimer in the template DNA. The experiment was done on a non-damaged DNA template in order to understand the DNA polymerase activity in damage bypass. Steady-state kinetic analysis of the deoxynucleotide incorporation indicates that the DNA polymerase has a low fidelity and low processivity. Here are the results from the experiment. The DNA polymerase was determined to have a low processivity. It could only incorporate a few nucleotides before dissociating. The low processivity limits its activity to reduce errors. The DNA polymerase was also determined to have a low fidelity. It had a frequency of about 10 to the negative 2 to 10 to the negative 3 of incorporating the wrong nucleotide. This was determined by measuring the steady-state kinetic assay of the correct and incorrect nucleotide incorporation. The highlighted region shows DNA synthesis by the polymerase from a single binding event. The next region shows the percentage of the DNA polymerase molecules incorporating nucleotides before dissociating. The highlighted region shows the incorporation of nucleotides across a template. The DNA polymerase was incubated with a primer template DNA substrate with increasing concentrations of the wrong and right deoxynucleotides. This region shows the quantification of the deoxynucleotide incorporation reaction. The rate of the incorporation is graphed as a function of deoxynucleotide concentration and was fit to the Michaelis-Menten equation.